Welcome back to Intro to Physical Anthropology. I'm your instructor, David Leitner, and today we're going to focus on the structure of DNA. What is this molecule? Why is it important? How is it made up? How does it do what it does? Well, we'll find out. DNA is a very long chain molecule that is formed into something called a double helix. That is, it looks like a ladder that somebody has twisted down the middle, right? As you can see on the far right here. Each of the steps of the ladder is composed of a nucleotide, or rather a pair of nucleotides. A nucleotide is simply a molecule that has a phosphate and a sugar called deoxyribose base. That's the backbone you see here, the blue and the yellow. And then it has something called a base. These base molecules come in two flavors. The first are pyrimidines. They are either cytosine, which we represent with a C, or thymine, which we represent with a T. Little side note, when we talk about protein synthesis, we're going to be talking about RNA. RNA uses the same code except for one exception. Thymine is replaced by uracil. There is no thymine in RNA. So, next we have the purines, and they are represented by adenine, which is A, and guanine, which is G. Okay. The neatest thing about this is that these bases can only connect in a very particular way. It's called the pairing rule. A and T always pair, or conversely, A and U. C and G always pair, but you never pair G and A, or C and A, or C and T. None of those others. These are the only combinations you can have. A and T, or C and G. That's it. And that little rule actually gives us a great deal of variety of different forms that we can actually encode information with. Here's another representation of the molecule here. You can see the P is the phosphate, D-deoxyribose, and that forms a backbone on either side of the ladder here. And here they've chosen to represent A and T as complementary shapes and C and G as complementary shapes to kind of represent the way that they only fit in one pattern. Okay. Now, what does DNA actually do? Well, it has several main purposes. The first is replication. Replication makes new DNA, okay? And it's sort of a four-step process. I'm going to run through it fast, and then we'll run through it slow, okay? The first thing that happens is the DNA uncoils, so it untwists, okay? And it starts to unzip at one end until it unzips completely in half. So whereas you had you had one double strand of DNA, now you have two single strands of DNA. Just unzips, just like a zipper. The next thing that happens is that each of those sides will serve as a template for the new DNA. Okay, so we talk about the mother DNA, or the mother strand, and we talk about the daughter strands here. The mother strand splits apart into the daughter strands, and then the daughters, using the pairing rules, attract other nucleotides, free-floating nucleotides in the nucleus to those sites to pair up. So they sort of build the other side, mirror image, piece by piece, until you now have two double strands. So here you can see the double strand mother strand of DNA splitting apart And then a little molecule called polymerase comes in and reads from one direction or the other, depending on which strand it's on. And it reads what nucleotide is at that one site, and it pulls in the complementary nucleotide, the one that pairs with it. And then it attaches it to the previous one, and it starts making a double strand that way. And here you have it. So you end up with 2 new strands. Each strand has a piece of the old strand in it. Okay. So enzymes, now this is a supremely oversimplified version of this process. There's a lot of stuff going on. There are a lot of other molecules other than polymerase that are involved in this. But they're all enzymes. They facilitate molecular reactions, biochemical reactions. Polymerase 1 was the first enzyme discovered that was associated with the process of replication, but there are others. Each of them controls a different aspect: the uncoiling, the separation, the reassembly, and a step of proofreading that happens after it's all together to make sure there weren't any mistakes made. When there are mistakes made, even with the proofreading, they sometimes miss the mistake, and if that makes it into the final double-stranded DNA, we call that a mutation. Okay, that's all a mutation is. It is a single letter change in the DNA sequence, and that's it. Because after that, every time that strand of DNA splits apart, it's going to continue replicating that mistake, right? Mutations are the only way new traits arise, okay? Brand new traits. Most mutations, however, are benign and have no effect on cell function whatsoever. You were born with 40 to 50 mutations that your parents did not have, okay? So you start off as a mutant. We all do. Other mutations might impact the cells, but we'll get more into that in later videos when we talk about mutation. For a mutation to be passed on to your offspring, it has to occur in the gametes. Mutations in somatic cells do not get passed on. I'm going to leave it there for today, and in the next video we will talk about the second function of DNA, which is protein synthesis. Take care of yourself. Hope you have a great day, and I'll see you soon.